Scientists from around the world shed their winter coats and met on the Sanford-Burnham Lake Nona campus in Central Florida to share their latest research into the origins of metabolic diseases.
I'm just telling you what.
Keynote speaker Sir Stephen O'Reilly of the University of Cambridge reminded everyone of the value of studying rare genetic diseases to illustrate mechanisms fundamental to more common metabolic disorders.
Showed a number of examples of individuals who had unusually severe forms of insulin resistance that we can understand and that might guide us to therapy for these unfortunate people. What we're discovering is many of the genetic variants that influence obesity actually do influence how the brain perceives food and how it wants to ingest food. So on the one hand, genetics is influencing this behavior. We are currently undertaking clinical trials with particular, we found a large number of people, there are about 50,000 people in Britain who are obese because they have a particular mutation called an MC4R. And in those, we're undertaking a trial with an American biotech company of a particular treatment targeted to those people. So we're doing it, one step at a time.
Sanford-Burnham's own Sheila Collins is studying ways to utilize brown fat to burn excess energy so that it is not stored as fat in the body.
So now we like to think about these brown fat cells as a cell that maybe we can use it to burn off excess calories and begin to shift the metabolic balance from continually storing energy and eventually leading to obesity and metabolic disease. And can we begin to either protect against that or reverse that process?
Donald McDonnell of Duke shared new insights into how breast cancer and cholesterol metabolism are linked together through the hormone estrogen.
But one of the things that's frequently forgotten is that about 20% of cancers are related to obesity. It's a modifiable risk factor, and that if we understand the mechanisms of which obesity impacts the development of cancer, we might be able to develop interventions that actually can mitigate some of the harm. And so the message for me is that diet and cancer are linked, and that by understanding these links, we may be able to do something. And now what the big finding of our study is something that people hadn't really contemplated before, and that is cholesterol. And circulating cholesterol itself is also a risk factor for breast cancer. So people had basically done epidemiological studies showing that, metabolic syndrome, for instance, increases breast cancer risk. But nobody had really drilled down to figure out what are the relative contributions of, say, triglycerides, glucose, or cholesterol to breast cancer incidence. And so the study that we have just recently completed have really shown that cholesterol is one of the culprits linking obesity, metabolic syndrome, and breast cancer risk.
Deborah Muoio of Duke has a theory on the relationship between mitochondrial fat overload in muscle and obesity-related diseases.
So we're very interested in how these lifestyle factors impact mitochondrial function, and then in turn, how does mitochondrial function impact things like the aging process and metabolic disease. And so what we're really trying to understand is, in the setting of overnutrition and obesity, how do the mitochondria respond essentially when they're force-fed nutrients that they really don't need? And so we've been using techniques referred to as metabolomics that allow us to measure hundreds and potentially even thousands of metabolites in a single tissue or sample. And many of those metabolites are derived from the mitochondria, and they can give us insight into how these organelles are responding to various physiological stresses and other environmental stresses.
Bruce Spiegelman of Harvard Medical School has made seminal contributions to the field of energy metabolism and obesity. His recent discoveries include two proteins that seem to mimic some of the benefits of exercise.
So we've identified two proteins, irisin and meteorin-like, which are independent molecules, but they both are secreted by muscle under conditions of physical activity and exercise. And they affect the adipose tissues in a way that makes them healthier and that reduces diabetes and obesity. It looks like irisin has more direct effects on adipose cells that turn on a thermogenic program, a program of what we call browning, making them more like brown adipose cells. And meteorin-like affects adipose tissue, but works indirectly and involves the immune cells in this response. The adipose tissue has many immune cells in it, and a lot of the action of meteorin-like is not directly on the fat cells, even though it winds up with the fat also being browned, having more thermogenic capacity, many of those actions are not necessarily directly on the adipose cells themselves. So the mechanisms of irisin and meteorin-like actually appear quite different, although the ultimate biology is similar.
Mitch Lazar of the Diabetes Center at the University of Pennsylvania is studying how metabolism is integrated with circadian regulation of sleep-wake cycles.
Well, we're very interested in metabolism and physiology and try to understand how things work in normal physiology and how that goes wrong in pathophysiology, especially with obesity and diabetes. And what we noticed was in brown fat in mice, the rhythm of the heat of the brown fat and of the body temperature was opposite to that of a molecule we've been studying for a long time, which is called reverb. And reverb is a transcription factor, member of the nuclear receptor family, and a strong repressor of transcription. And so we made the hypothesis that it was driving the higher body temperature, and what we did was we basically made mice genetically lacking reverb, and they were better able to tolerate a cold temperature stress, and they also had a reduction in their dip when they're sleeping of body temperature, again consistent with the idea that reverb was actually playing a role in that circadian rhythm.
Robert Gersten of Harvard Medical School is pioneering the use of metabolic markers in the blood to diagnose current disease, as well as to predict the likelihood of disease development in the future.
I think that these new techniques for profiling lots of chemicals in the blood are really going to redefine human metabolism. I'm not saying our work in any way, shape, or form, but I think it's beginning to suggest as proof of principle that by looking at a broad swath of chemicals in the blood, we're ultimately going to redefine human physiology. Again, just what we're doing, I want to underscore, is a very early step in that process, but doing things in an unbiased way with these new types of tools really holds the potential of changing the way we think about physiology.
Research findings presented throughout the symposium underscored that investigating metabolism is crucial for understanding and treating a variety of diseases.